Hello guys, welcome back to my channel. In this video we'll be doing the second chapter of biotechnology, that is biotechnology and its applications. So please subscribe my channel and click on the bell icon. Let's get started. This is chapter 12, biotechnology and its applications. In the previous video we have already done the first chapter in two parts. So now we will do with this second chapter. So biotechnology, as you would have learned from the previous chapter, essentially deals with the industrial scale production of biopharmaceuticals and biological using the genetically modified microbes, fungi, plants and animals. This application of biotechnology includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. So, please highlight this on your NCERT. Uh, Wait, just a minute. Okay, so its applications include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. So now, the three critical research areas of biotechnology are providing best catalyst in the form of improved organisms, usually a microbe or pure enzyme, then creating optimal conditions through engineering for catalyst to act, and downstream processing technologies to purify the protein or the organic compound, which we have already seen in the previous chapter. In previous chapter we have already done the downstream processing, and before that the extraction of protein and the thing like organic compound was done using the bioreactors. So now in this chapter we'll do the remaining things like different types of organisms which are genetically modified, their use, and what are its benefits or advantages for the humans. So let us now learn on how human beings have used this biotechnology to improve the quality of human life, especially in the field of food production and health. Health means some organisms they are modified, so they act as a model for studying different types of symptoms of different diseases like Alzheimer's disease, cystic fibrosis, etc. And some of them are also modified so that vaccine safety can be tested on them, like mice. Even monkeys are used to test the safety of vaccine before they are used on humans. So let us take a look at three options that can be thought for increasing the food production. One is agrochemical based agriculture using the fertilizers, pesticides and all agrochemicals. Then second is organic agriculture, in which we use the organic things like manure, then other plant waste, animal waste, and we keep the microbes at minimum level, and we don't kill them, or we keep them at manageable level. Then third is genetically engineered crop based agriculture, which we will learn in this chapter. So the green revolution succeeded in tripling the food production, but yet it was not enough to feed the growing human population. The increased yields have partly been due to the use of improved crop varieties, but then mainly due to use of better management practices and use of agrochemicals, that is fertilizers and pesticides. The three point which are very important and which played great role in the green revolution are increased those 3 points they helped in increasing the yield, those points are use of improved crop varieties, then better management practices and use of agrochemicals, that is fertilizers and pesticides. However, for farmers in developing world, agrochemicals are often too very expensive, and further increase in yield and existing varieties are not possible using the conventional breeding. So जो conventional breeding methods है उससे increase in yield it's not possible to cope up with this increasing demand. So is there any alternative path that our understanding of genetics can show so that our farmers may obtain a maximum yield from their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on animals are on the environments are reduced? Use of genetically modified crops is a possible solution. That is, we are now discussing the third point. After doing organic agriculture, we are now heading towards the genetically engineered crop based agriculture. And now the plants, fungi, and bacteria, which are whose genes have been altered by manipulation are called as genetically modified organisms or genetically modified crops in case of plants. The plants have been useful in many ways. The genetic modification in plants, they have made crops more tolerant to abiotic stresses like cold and the salt, heat, etc. The salt means the salinity. Then they've reduced reliance on chemical pesticides, the pest resistant crops, and they have helped to reduce the post harvest losses. Even they have helped to increase the efficiency of mineral usage by plants. That is, this prevents early exhaustion of fertility of soil. And in nutritional value of food, that is golden rice, like example is vitamin A enriched rice, also called as the that is beta carotene enriched rice, also called as the golden rice, are few examples. Genetically modified organisms or genetic modification has been used to create a tailor-made plants to supply alternative resources to the industries in the form of starches, fuels and pharmaceuticals. Like there are many other products or many other raw materials required in the industries which we get from plants, and they have been produced using this genetic modification. Some application of biotechnology in agriculture that we will study in detail are production of pest resistant plants which could decrease the amount of pesticide used. That is, we have already seen that the genetic modification has made plant less dependent on the chemical pesticides and all, because production of pest resistant plants which could decrease the amount of pesticide used. And bt toxin, which is produced by a bacterium called bacillus thuringiensis, we say BT for short, and BT toxin gene has been cloned from the bacteria and has been expressed in plants to provide resistance to the insects without the need of insecticides. In fact, created a biopesticide. Examples are BT cotton, BT rice, then BT corn, tomato, potato, and soybean. These are the examples of the plants which are resistant to the certain pests like bt cotton, bt rice, tomato, corn, etc. So we'll first look at this bt cotton, which is one of the examples. Bt cotton, some strains of bacillus thuringiensis bacteria, they produce proteins that kill certain insects which are such as the lepidopterans, then Coleopterans. The lepidopterans examples are tobacco budworm, armyworm, coleopterans like beetles, and dipterans like flies and mosquitoes. Bacillus thuringiensis forms a protein crystal during a particular phase of their growth. मतलब जो प्रोटीन बनता है, it is formed only during that particular phase of the growth and not at any other time. So it's like a seasonal breeder, or it is seasonal, or you may say it is produced only during that particular stage. And secondly, it is insect group specific. It is not the particular protein is specific for that particular insect. Okay, so these crystals, they contain a toxic insecticidal protein. I hope you are marking on your NCERT book at the same time, because these points are very important and they might come for boards or even for NEET. So why does we'll go to the next para now. Okay, no, first we'll look at this question. Why does this toxin not kill the bacillus, the bacteria which produces this toxin? Why is that toxin it's not able to kill this bacillus? Actually, the bt toxin protein or the bt toxin which is produced, it exists as an inactive protoxin, so it is produced as an inactive protein. Like there are certain enzymes, or no, mostly all the enzymes in our stomach like pepsinogen, trypsinogen, they are produced in inactive form. This pepsinogen is inactive form, trypsinogen is inactive form, prorenin is inactive form. So they are in inactive form, so they are non-functional. So they have to be converted first into a active form, so that they are produced in inactive form, so that they don't affect the neighboring cells or the cells which produce them. Similarly, this toxin or this toxic protein is produced in inactive protoxin form, so that it doesn't affect this bacteria. So but once the insect or the pest ingests the inactive toxin, it gets converted into an active form of toxin. It's because of alkaline pH of the gut which solubilizes the crystals. The pH inside the gut of this insect or pest is alkaline, and this alkaline pH is favorable for its conversion to a active form. So that's why it gets converted into active form, and this activated toxin it binds to the surface of the midgut epithelial cell. जो उनके midgut में epithelial cells होते हैं, it binds to those midgut epithelial cells and then create pores which cause cell swelling, lysis and eventually cause the death. So जो cells होते हैं midgut epithelial cells उनमें वो pores create करते हैं, swells then swellings आती है cells में and lysis हो जाता है, eventually they die. So specific Bt toxin genes, these toxin genes which are present in the bacteria were isolated from the Bacillus thuringiensis bacteria and incorporated into the several crop plants such as cotton. Like in Bt cotton, they are incorporated into cotton. In the case of Bt rice, in rice. Similarly, so we here in this case we are looking, we are considering the case of cotton. So this gene was incorporated into this was isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes it depends upon the crop and the targeted pest. So I've already told you that this is insect group specific. So it depends upon the crop and the targeted pest, as most of the Bt toxin genes are insect group specific. So just mark this. The choice of gene depends upon the crop and targeted pest, as most of the Bt toxins they are insect group specific, and it is coded by a gene called cry gene. It is coded by the cry gene. There are a number of them. For example, the proteins or those toxic proteins or Bt toxin, you can say any one of them. They are encoded by a gene called cry gene, and those proteins which are encoded by cry1AC and cry2AB gene, they control cotton bollworms. So they are effective against cotton bollworms, whereas the cry1AB controls cotton borer. So cry1AC and cry2AB control cotton bollworms, and cry1AB controls corn borer. So I hope you have understood this part and you have also marked this on your NCERT. So now we'll go to the next part, that is pest resistant plants. We have already seen one of the pest resistant plant, that is the Bt cotton, which is made by isolating the Bt toxic gene, that is the cry gene, from a Bacillus thuringiensis, and then it was incorporated into the cotton, and they are also insect group specific. So it depends on the type of crop and the insect. Okay, so the pest resistant plants, several nematodes they parasitize a wide variety of plants and animals, including the human beings. A nematode called Meloitygyne incognitia infect the roots of tobacco plant and cause great reduction in the yield. So जो रूट्स होते हैं टोबैको प्लांट्स में, उनमें जो वो पैरासाइट है मेलोइडीगाइने इनकॉग्निशिया, वो इन्फेक्ट करता है उसको, and then it causes reduction in the yield, maybe because it interferes in the metabolism or something else. So a novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, that is RNAi or RNA interference. RNA interference takes place in all the eukaryotic organisms as a method of cellular defense. So question can come like, RNA interference takes place in all the eukaryotic organisms. Give reason for that. The reason is it acts as a method of cellular defense, and the method involves silencing of a specific mRNA due to the complementary double-stranded RNA molecules that binds to it and prevents the translation of mRNA. So this prevention of this mRNA is called its silencing. Okay, this method involves silencing of specific mRNA due to complementary double-stranded RNA molecules that binds to it and prevents the translation of mRNA, causing its silencing. This is called RNA interference. The question is, from where does this double-stranded RNA comes? We know that mRNA is already present, but this double-stranded RNA comes from outside. or it should be inserted, or it may enter through a viral infection or any other thing. So here they have given that this source of RNA, that is complementary RNA, the double-stranded RNA, could be from an infection by viruses having RNA genome or the mobile genetic elements called transposons that replicate via RNA intermediate. Using the agrobacterium vectors, nematode-specific genes were introduced into the host plant. In the previous chapter, we have already seen that Agrobacterium tumefaciens, it acts as a uh, uh, pathogen of dicot plant, but at the same time, it has been disarmed, and now it is no more pathogenic. The modified one is no more pathogenic, and it is used as a vector. So using for in case of plants. So these Agrobacterium vectors, using this vector, a nematode specific gene were they were introduced into the host plant, and this introduction of DNA or the gene was such that it produced both sense and antisense RNA in the host cell. Okay, so this sense and antisense RNA being complementary to each other, they formed a double-stranded RNA, and they initiated this RNA interference and caused the silencing of this mRNA. What is RNA interference? RNA interference is nothing but the double-stranded RNA. It binds to the mRNA and prevents its translation. This is called RNA interference. So, from where does this double-stranded RNA comes? This comes from either from a viral infection having RNA genome or the mobile genetic elements which are there. So, but in case of this artificially initiated RNA interference, using the agrobacterium, we have introduced the gene that produced both sense and antisense RNA, and sense and antisense RNA being complementary to each other, they form the double-stranded RNA, and this we require this double-stranded RNA to cause the silencing. It prevents translation of mRNA, and this is called silencing of mRNA. So here they have already given in the previous chapter, previous para, that using this RNA interference, this there is silencing of specific mRNA due to the complementary double-stranded RNA molecules that binds to it and prevents the translation of mRNA. So the double-stranded RNA we have already got, and that initiated the RNA interference and silenced the specific mRNA of that nematode. The consequence was that the nematode or that nematode parasite, it could not survive in the transgenic host expressing specific interfering RNA, and the transgenic plant therefore got protected from the parasite. In this way, we have made plants resistant to the certain kinds of pests or certain insects. They are highly group specific. If you transfer a gene against a particular insect, it may not work for other insect, but it is highly specific for that particular insect. Like we already seen the cry gene, it has many different types of cry genes like cry1ac, cry2ab. They are specific for the cotton bollworms, whereas the cry1ab is for corn borers. Okay, so in this image you can see clearly that the roots of tobacco plants, they are infected, and in figure B there are no such infections seen. So in the second figure there is RNA interference, that is transgenic plant roots 5 days after the deliberate infection of nematode, but it got protected through the novel mechanism which is present in all the eukaryotes as a method of cellular defense. So this is now a very important part of this chapter, and most of the questions come from this part also, because this is also very important, and this is, I would say, upcoming or growing field in future, and it has bright future. And the now all those vaccines and other products and drugs which are on which the research is going on is it is all based on the biotechnological applications in medicines. So we'll start this para now. The recombinant DNA technologies have made immense impact in the areas of healthcare by mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological responses, as is common in case of similar products obtained from the non-human resources or sources. At present, about 30 recombinant therapeutics have been approved for human use all over the world, and in India, only 12 are of this are presently being marketed. First point is recombinant DNA technologies have made immense impact in the area of healthcare by mass production of safe and more effective therapeutic drugs like vaccines and all. Then in second point, it says that recombinant therapeutics, they do not induce unwanted immunological responses, as is common in case of those isolated from non-human resources. Like uh, if certain chemicals or hormones, if they are isolated from non-human resources, then they might induce some immunological responses and they might cause allergies. But recombinant DNA technologies, this recombinant DNA therapeutics, they do not induce unwanted immunological responses. And at present, 30 recombinant therapeutics have been approved for human use all over the world. And in India, only 12 of these approved recombinant therapeutics are presently being marketed. So these 4 points are very important. Now we'll go to the next part, that is genetically engineered insulin. Management of onset diabetes is possible by taking insulin at regular time interval. You must have seen adults or those diabetic patients taking insulin. So what would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use this insulin from other animals. And this was earlier, they used to extract this insulin from the pancreas of slaughtered cattle and pigs. So here they have given it, insulin for diabetes was earlier extracted from the pancreas of slaughtered cattle and pigs. But insulin from such animal sources or non-human sources though caused some patients to develop allergies or other types of reactions to the foreign protein. So, insulin, it consists of two short polypeptide chains, chain A and chain B. The insulin has two polypeptide chains, short polypeptide chains, chain A and chain B. They have given in this diagram. These are the two chains, chain A and chain B here. And those chain A and chain B are linked together by a disulfide bridges. This is the bridges made up of sulfur, disulfide bridges. In mammals, including the humans, insulin is synthesized as a prohormone, inactive hormone. Just like the Bt toxin protein, which is produced as a protoxin, in mammals, including humans, insulin is synthesized as an inactive or a prohormone like proenzymes, and the prohormone needs to be processed before it becomes fully matured and functional hormone. So this inactive prohormone contains an additional or an extra stretch called C peptide. So already we know that there are two polypeptide chains, short polypeptide chains, that is chain A and chain B. But in immature or those insulins which are just produced, that is in inactive form or prohormones, they contain an additional or an extra stretch of peptide or a polypeptide, that is C peptide. This C peptide is present between chain A and chain B. Chain A and chain B are they are linked by disulfide bridges, but in mature insulin, this C peptide is not present. But when it is in prohormone form, it has additional or this extra stretch is present between chain A and chain B, and this is called a C peptide. So in this diagram, they have shown the gray color. The A is the blue color is peptide A, that is the A chain A, and the green is chain B or a B peptide. But in inactive one, the first one, proinsulin or a prohormone, there is a green part. There is a gray part between A and B. Between blue and green, there is a gray part. This is called a C peptide, and this C peptide is not present in the mature form. So this C peptide, which is present in immature one, is not present in a mature insulin and is removed during the maturation into the insulin. The main challenge for production of insulin using this recombinant DNA techniques was getting the insulin assembled into a mature form. So the main problem was कि जो insulin रिकॉम्बिनेंट डीएनए टेक्नोलॉजी से फॉर्म हो रहा था, उसको मैच्योर फॉर्म में लाना वाज अ चैलेंजिंग. And in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to chain A and chain B of the human insulin and introduced them in a plasmid of E. coli, that is Escherichia coli bacteria, to produce insulin chains. And these chains, that is the polypeptide A and B, these A and B chains were produced separately, extracted, and then combined by creating the disulfide linkages to form a human insulin. So I'll just revise these points. The first point is earlier insulin was extracted from non-human sources, uh, like the pancreas of slaughtered cattle and pigs. But this insulin, though caused some patients to develop allergies or other type of reactions, it was used. And the insulin, it consists of two polypeptide chains, that is it has chain A and chain B, but this is in mature form. But in mammals, including the humans, this insulin is produced as a prohormone, that is inactive or less functional or non-functional hormone, that is prohormone, and it has extra stretch of peptide called C peptide present between this A peptide and B peptide. A peptide and B peptide, or A and B chains of insulin, are linked together by disulfide bridges, and in this is in mature form. But when it is produced as a prohormone, besides this chain A and chain B, there is a C peptide present between chain A and chain B, and there are disulfide linkages also present between the chain A and chain B. So in mature form, this C peptide is not present. It is removed during the maturation into the insulin, maturation of proinsulin into insulin. Later, the Eli Lilly company, American company, it produced two DNA sequences called chain A and chain B corresponding to the chain A and chain B of insulin. They were then introduced into the plasmid of E. coli, and they produced this chain A and chain B separately. They were extracted, then recombined by creating the disulfide bonds or disulfide linkages to form a mature insulin. Now there is very important part, that is the gene therapy. So if a person is born with hereditary disease, okay, which can a corrective therapy be taken for such a disease? A gene therapy is an attempt to do this. So if a person is born with a hereditary uh, disease, a corrective therapy can be given for such disease through gene therapy. So this gene therapy is what? Gene therapy is nothing but it is actually a collection of methods that allow a correction of a gene defect that has been diagnosed in a child. So this gene therapy is a collection of methods. It is not a single process or a single method. It is a collection of methods that involves the. This gene therapy is a collection of methods that allows correction of gene defect that has been diagnosed in a child or an embryo. Here genes are inserted into person's cell and tissues to treat a disease, and correction of genetic defect involves the delivery of normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. So यहाँ पे जीन्स इंसर्ट की जाते हैं, जो फंक्शनल जीन्स हैं वो हम इंसर्ट करते हैं. Those inserted genes are functional and they take over the function of non-functional gene, or they compensate for the non-functional gene. First clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This is very important line. Please mark this line because this might come for NEET and even for boards. In which year the first clinical gene therapy was given? It was given in 1990, and to whom? To a 4-year-old girl. And what was the problem? The problem was adenosine deaminase deficiency. Actually, I'll tell you what happens. Adenosine deaminase gene is present, and this gene is responsible for producing the adenosine deaminase enzyme. And this enzyme is actually required for maturation of these lymphocytes, or it is involved in the uh, mm, immunity. It is involved in defense mechanism. provide immune response. So what happens is when this adenosine deaminase gene is not present, or when this becomes non-functional, there is no production of adenosine deaminase enzyme. So the person suffers from uh, its immunity becomes very low, and it suffers from a syndrome or different types of diseases. So it is called severe combined immune deficiency. So the person's immunity becomes very low, and becomes that person is he becomes highly sensitive to antigens present in the environment, or many other bacteria, then other pathogens. They can easily infect the person, and it is very fatal. Like person can easily get tuberculosis, or even other common cold can also become very severe for that person. So this enzyme is crucial actually for immune system to function. The enzyme, that is adenosine deaminase enzyme produced by adenosine deaminase gene, is crucial for immune system to function, and this disorder is caused due to the deletion of the gene for adenosine deaminase. So when the gene is deleted, or when it is non-functional, this disorder is caused. And in some children, this adenosine deaminase deficiency can be cured by bone marrow transplantation, while in others, it can be treated by enzyme replacement therapy, in which a functional adenosine deaminase is given to the patient by injection. So, in few children, it can be treated with the help of this bone marrow transplantation. Not in all. In few cases, it might be successful, but it is not complete cure. So in some children, adenosine deaminase deficiency can be cured by bone marrow transplantation, but there is no permanent cure for this. So in others, it can be treated by enzyme replacement therapy, in which functional adenosine deaminase enzyme is given to that patient or that children who are having the adenosine deaminase deficiency by the injection. So this adenosine deaminase enzyme is given to that person or that child having adenosine deaminase deficiency by injection, and this is called enzyme replacement therapy. But the problem with both of these approaches, uh, that they are not completely curative. Even if you do the bone marrow transplantation or enzyme replacement therapy, they might not be 100% successful, or they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of patients are grown in a culture outside their body. First step is take the lymphocytes out of person's blood and grow them in culture outside the body. Then functional adenosine deaminase cDNA using retroviral vector is introduced into these lymphocytes, and which are subsequently returned to the patients. So एडिनोसिन डीएमिनेज cDNA जो है वो रेट्रोवायरल वेक्टर से इंट्रोड्यूस करते हैं लिम्फोसाइट्स में, बिकॉज इन वी हैव ऑलरेडी सीन दैट इन केस ऑफ एनिमल्स वी यूज द रेट्रोवायरस, इन केस ऑफ प्लांट्स एग्रोबैक्टीरियम. So since this happens in animals, we use the retrovirus. So now we have already inserted this adenosine deaminase cDNA into the lymphocytes, and now this is returned to the patient. But since these cells are not immortal, the patient requires periodic infusion of genetically engineered lymphocytes. उसके lymphocytes जो हैं उनमें वो जीन नहीं है, that is already deleted, and it is suffering from adenosine deaminase deficiency. So that's why what we have done, we have removed those lymphocytes from its blood, grown them in culture medium, and inserted cDNA, adenosine deaminase cDNA, using retrovirus. After that, we return those lymphocytes were returned to the patient, but since they are not immortal, they require periodic infusion of such genetically modified or genetically engineered lymphocytes. So we don't know exactly which is 100% successful method, but if the gene isolated from the marrow cell producing adenosine deaminase is introduced into the cells at early embryonic stages, it could be a permanent cure. Okay, so if the gene is uh, if the gene is isolated from the marrow cells producing adenosine deaminase, जो adenosine deaminase प्रोड्यूस करते हैं marrow cells, उनमें अगर वो जीन इंसर्ट कर दी, sorry, I made a mistake here. If the gene is isolated from the marrow cell, जो marrow cells में से अगर gene isolate कर दी producing the adenosine deaminase and अर्ली एम्ब्रियोनिक स्टेजेस पे अगर सेल्स में हमने वो इंट्रोड्यूस कर दी, देन इट कुड बी अ परमानेंट क्योर, बट वी डोंट नो, वी आर नॉट वेरी 100% श्योर अबाउट इट. So I'll again repeat those points. And first step, what we did, the bone marrow transplantation, it can be done in few children, but it's not completely curative. Then again, enzyme replacement therapy, in which functional adenosine deaminase cDNA using retrovirus is introduced into the patient or that person, but it's not completely curative. Then we did the gene therapy. The first step is take the lymphocytes out of the blood, grow them in the culture medium, then using adenosine, then using retrovirus vector, introduce the adenosine deaminase cDNA. Then subsequently they are returned to the patient, but since they are not immortal, they require periodic infusion. So this is also not permanent cure, but gene isolated from the marrow cells producing adenosine deaminase if it is introduced into the cells at early embryonic stages, then it could be a permanent cure. Okay, I hope this is clear and you must have understood this now. Molecular diagnosis. You know that for effective treatment of disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis like serum and urine analysis, etc., early detection is not possible. You must have seen, or you must have even given your serum or urine for such analysis. So these are conventional methods or conventional practices. These are conventional methods of diagnosis, but early detection using this method is it's not possible. So that's why the recombinant DNA technologies or polymerase chain reaction, even the enzyme linked immunosorbent assay, that is ELISA, are some of the techniques that serve the purpose of early diagnosis. These are more advanced and they are all based on the biotechnology applications. So presence of a pathogen like bacteria, virus or fungi, etc., it is normally suspected only when the pathogen has produced a disease symptom, and then we use the conventional methods of diagnosis, but they don't help in early detection. But by this time the concentration of pathogen is already very high in our body. However, very low concentration of a bacteria or a virus at a time when the symptoms of this disease are not yet visible can be detected by amplification of this nucleic acid by PCR, polymerase chain reaction. We have already done this part in the previous chapter, that is the amplification of gene using the polymerase chain reaction, PCR. There are three steps. One is denaturation, the first step, then annealing using the primers, and the final step is extension using the thermostable DNA dependent DNA polymerase, that is the Taq polymerase. These are the three steps. Using this we do the amplification of the nucleic acid. PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect the mutations in the genes in suspected cancer patients too. So it is powerful technique to identify many other genetic disorders. So एक तो हम वो HIV डिटेक्ट करने में यूज़ करते हैं, एंड सेकेंडली वी यूज़ दोज टेक्निक्स और दिस PCR to detect mutations in the genes in case of suspected cancer patients. Okay, a single stranded DNA or RNA tagged with a radioactive molecule is called a probe. What is a probe? A probe is a single stranded, it could be a DNA or RNA tagged with a radioactive molecule is called a probe. That is tagged with a radioactive molecule. It is single stranded. Remember these 3 points. The probe is single stranded, it could be a DNA or RNA, and third is it is tagged with a radioactive molecule. This is probe, and it is allowed to hybridize a complementary DNA in a clone of cells, followed by detection using autoradiography. So the clone having mutated gene will hence not appear on the photographic film. It's because the probe will not have complementarity with the mutated gene. क्या होता है मैं बताता हूँ. जब प्रोब जो सिंगल स्टैंडर्ड डीएनए और आरएनए होते हैं टैग्ड विद रेडियो एक्टिव मॉलिक्यूल, व्हेन वी मिक्स देम विद दिस म्यूटेटेड जीन being complementary, they bind with those sorry when we mix them with these normal genes, they bind with those genes and then they appear on the photographic film using the autoradiography. But when there is mutated gene, this this probe, it will not show the it will not have a complementarity with those mutated genes. So that's why the mutated genes, they will not appear on the photographic film or the using the autoradiography. So this is simple technique to detect the mutations in the suspected cancer patients. Then there is ELISA, that is enzyme linked immunosorbent assay. It is based on the principle of antigen and antibody interaction. These are again of two types. So in NCERT they have not given in detail, but ELISA is of two types. It is based on antigen antibody interaction. Infection by pathogens can be detected by either presence of antigens or by detecting the presence of this antibodies synthesized against the pathogens. So दो टाइप के होते हैं. One is, so the main function is it detects the interaction. It is based on the principle of interaction between antigen and antibodies. First is the one type is detecting the antibodies produced against this pathogen, and the second is detecting the antigens, presence of antigens of those viruses or the pathogens. Now the next part is transgenic animals. This is very easy. I hope you will do this. It needs no explanation. It is just you can read it and just mark the important things. It's very easy. So I will not do this part. You have to read it on your own. Okay, so thank you guys for watching this video.